Hey, how's it going, everyone? This is FixerMed. Welcome back to my high yield immunology review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 3 of a multi-part video series diving deep into the discipline of immunology in a comprehensive but high yield manner. So, without further ado, let's go ahead and get started on today's content, which should be a review of lymphocytes. So I'd like to start off talking about natural killer cells. These are the main constituent of innate immunity. They're triggered by IL-12. Natural killer cells alongside B and T cells constitute the trio of lymphocytes originating from lymphoid progenitors. Natural killer cells eliminate virus-infected and cancerous cells by detecting alterations in cell surface protein expression, such as reduced MHC1 expression. CD16 on natural killer cells facilitates antibody-dependent cell-mediated cytotoxicity. Antibodies bind antigen via their FAB regions, exposing FC regions outward. CD16 on natural killer cells bind to FC regions, initiating cross-linking. Cross-linking prompts natural killer cells to release granzymes, granulysins, and performance, inducing apoptosis in the target cell. So that's what we got for natural killer cells. Let's go ahead and move on to the next type of cell we'll be discussing. We're going to be talking about B cells next. B cells are the principal agents of humoral immunity. These differentiate into plasma cells secreting antibodies. B cells undergo somatic hypermutation and affinity maturation, enhancing antibody specificity for antigens. Germinal centers and lymph nodes and spleen foster B cell proliferation and differentiation. B cells function as antigen-presenting cells, capable of engulfing extracellular pathogens and presenting them to CD4 plus T cells via MHC2. Let's go ahead and move on to the final type of lymphocyte we'll be covering today, and those are T cells. The two crucial subsets to focus on will be CD8 plus and CD4 plus. CD8 plus cytotoxic T cells combat intracellular pathogens, mainly viruses and some bacteria, by binding to MHC1 on nucleated cells and inducing apoptosis. CD4 plus T cells encompass various subtypes. Th0 is a precursor that can differentiate into Th1 and Th2. Th1 primarily activates macrophages, while Th2 predominantly stimulates B cells for antibody production. Now that we've covered all of the lymphocytes, let's go ahead and move on to some questions. So, which of the following best describes the function of natural killer cells and the immune response? Alright, I think I gave you guys enough time there. If you need more time, please feel free to pause the video. Otherwise, moving on now. Natural killer cells play a crucial role in innate immunity by identifying and eliminating virus-infected and cancerous cells. They recognize these abnormal cells through alterations in cell surface protein expression, such as reduced MHC1 expression. Once identified, natural killer cells induce apoptosis in the target cells, contributing to immune surveillance and defense against infections and malignancies. Therefore, the correct answer is C. Natural killer cells detect and eliminate virus-infected and cancerous cells based on altered surface protein expression. Let's go ahead and see why the other answer choices are incorrect. IL-10 does not stimulate natural killer cells. It is IL-12 that triggers their activation, as we covered earlier. Additionally, natural killer cells primarily target intracellular pathogens, not extracellular ones. Natural killer cells are part of innate immunity, not adaptive immunity, and they do not undergo somatic hypermutation. While natural killer cells can engage in phagocytosis to some extent, their primary mechanism of action is through inducing apoptosis in target cells, not direct phagocytosis. Natural killer cells do not directly release antibodies. Instead, they facilitate antibody-dependent cell-mediated cytotoxicity through CD16 receptors, which bind to antibodies attached to target cells, leading to the release of cytotoxic molecules and the induction of apoptosis. All right, let's go ahead and move on to the next question. Which of the following statements best describes the role of B cells in the immune response? Alright, I think I gave you guys enough time there. If you need more time, please feel free to pause the video. Otherwise, moving on now. B cells undergo somatic hypermutation and affinity maturation processes within germinal centers, leading to the production of antibodies with increased specificity for antigens. This mechanism allows for the generation of high-affinity antibodies that can effectively neutralize pathogens and contribute to the adaptive immune response. Therefore, the correct answer is C. B cells undergo somatic hypermutation and affinity maturation to enhance antibody specificity for antigens. Let's go ahead and see why the other answer choices are incorrect. B cells primarily target extracellular pathogens, not intracellular ones, and they do not induce apoptosis in infected cells. This is a function primarily carried out by cytotoxic T cells. B cells do not differentiate into CD8 plus T cells. Instead, they differentiate into plasma cells that secrete antibodies. CD8 plus T cells are a separate subset of lymphocytes involved in cell-mediated immunity. While B cells can interact with macrophages and enhance their activation, they do so primarily through antigen presentation and cytokine release, not by directly activating macrophages through cytokines. While some phagocytic activity may be observed in B cells, their primary role is antibody production and antigen presentation, rather than direct phagocytosis of pathogens. Alright, now that we got that sorted out, let's go ahead and move on to the next question. Which of the following statements accurately summarizes the role of CD4 plus T cells in the immune response? Alright, I think I gave you guys enough time there. Pretty first order question. Gonna go ahead and move on now. If you need more time, please feel free to pause the video. Moving on now. CD4 plus T cells can differentiate into different subtypes such as Th1 and Th2. Th1 cells primarily activate macrophages, enhancing their ability to eliminate intracellular pathogens. Th2 cells, on the other hand, stimulate B cells to produce antibodies, therefore contributing to the humoral immune response. Understanding the distinct functions of these CD4 plus T cell subtypes is crucial for comprehending immune regulation in response to different types of pathogens. Therefore, the correct answer here is D. CD4 plus T cells differentiate into various subtypes, including Th1 and Th2, which play distinct roles in immune regulation. Gonna go ahead and move on to see why the other answer choices are incorrect. CD4 plus T cells primarily assist in immune regulation and do not directly target and neutralize pathogens. Their main role is to regulate the activity of other immune cells. CD4 plus T cells do not differentiate into cytotoxic T cells, CD8 plus T cells. CD4 plus T cells are primarily involved in coordinating immune responses rather than directly killing infected cells. While CD4 plus T cells can influence macrophage activation, they do so primarily through cytokine signaling and not by enhancing macrophage phagocytosis directly. CD4 plus T cells do not directly produce antibodies. They stimulate B cells to differentiate into plasma cells, which then produce antibodies as part of the humoral immune response. Guys, that will do it for our basic introduction to lymphocyte functions, etc. We'll be going into deeper detail onto these lymphocytes in the videos coming forward, or I should say going forward and coming out soon. But yeah, if you guys have any questions, comments, or concerns, please feel free to leave them in the comment section below. As always, if you like this video, please feel free to leave a like and subscribe for more content like this. It really helps the channel out a lot. And this is FixerMed signing off. Be sure to have a great day, good luck studying, and goodbye, everyone.